Hi, it's Miss Barnhart, and today we're going to talk about plant regulation and control. In my classroom this year, we planted tomato seeds that had been in a simulated space-type environment to see if there were any differences in the way the plants germinated and grew. As those tiny seeds germinated, they utilized all of the food supplies stored in their cotyledons, and we saw them grow towards the light. One group inadvertently blocked the light, and we saw the stems of these plants bent the following day. These plants, like the tree here, have regulatory processes that allow them to respond and survive. Like animals, plants use chemical messengers to regulate cell activities. A hormone is a chemical that is produced in one area of an organism's body and has effects on a distant target cell. Some botanists prefer the term plant growth regulator rather than hormone, since plants do not have circulatory systems and some of these chemicals are not biologically made. The idea that plants are chemical messengers all stem from studies on how plants respond to light, or phototropism. Tropisms are growth response in plants. Shoots exhibit positive phototropism because they grow towards sunlight. In 1890, Charles and Francis Darwin removed and covered grass coleoptiles to determine what part sensed the light. Notice that in the control, the shaded side of the stem had cells that grew longer, causing the curvature. Here's an illustration of some of Darwin's experimental findings. They noted that phototropism only occurred when the tip was illuminated, so they concluded that the tip must be responsible for sensing light. These findings were expanded by Boyson and Jensen as they concluded that the bending only occurred when the tip was separated by a permeable barrier, suggesting that the signal was a mobile chemical. In 1926, Wimp further expanded upon these studies by isolating the chemical messenger on the agar block. When he grew the plant with a decapitated tip in the dark, no bending occurred. No bending occurred on the plants that had a plain agar cube placed upon them and then placed in the dark, or on those that had the agar cube with a chemical messenger centered over the tip. However, they noted that when the agar cube was placed off-center, the plants curved away from the side with the cube, suggesting that the chemical caused increased cell growth. If the plants grow longer on the shaded side, this will cause the plant to bend towards light. The signal molecule was named auxin. Studies in plants other than grass suggested that illumination on one side caused growth inhibitors to concentrate on the lighted side of the stem. But the discovery of auxin led to the search for other plant hormones. The major plant hormones and their functions are as follows. Auxin is found in the embryo of the seed, in young leaves, and in the apical meristem. In addition to stimulating cell elongation and being involved in phototropism, auxin also plays a role in gravitotropism, apical dominance, and vascular differentiation. It inhibits abscission prior to the formation of the abscission layer. It stimulates the synthesis of ethylene in fruit formation. Additionally, auxin induces adventitious roots on cuttings. Cytokinins are made in roots and transported to other organs. Cytokinins stimulate cell division and reverse apical dominance. Gibberellins are made in the meristems of apical buds and roots, in young leaves, as well as in the embryo. These hormones stimulate shoot elongation and stimulate bolting in flowering and biennials. Gibberellins are also involved in the metabolic processes that lead to seed germination. Another hormone, ethylene, is produced in the tissues of ripening fruits, nodes of stems, and in aging leaves and flowers. Ethylene stimulates fruit ripening as well as the aging of leaves and flowers and the abscission of leaves. Ever heard the saying, one bad apple spoils the whole bunch? That's because of ethylene. Abscisic acid is found in leaves, stems, and green fruit and plays a major role in stomatal closure. Can you see that all hormones, with the exception of abscisic acid, stimulate growth? Have you ever wondered why plants only flower at certain times of the year? The purpose of flowering is to allow pollination, fertilization, and seed dispersal. Since flowers should only bloom when a suitable pollinator is abundant, these species will show seasonal population shifts. Long day plants bloom in the late spring and summer when the days are longer, while short day plants bloom in the late fall and winter when the days are shorter. Let's take a look at two different plants, the iris and the chrysanthemum. The iris is a long day plant since it flowers in the summer, but not in the fall. The chrysanthemum flowers in the fall but not the summer. It's a short day plant. The control of flowering is achieved through a process called photoperiodism. The critical factor is actually not the length of the day, but the length of the night, or the dark period. This is shown here. This box represents the critical night period. You'll see that when the night is longer than a certain length, your long day plant does not flower because the night is too long. In a short day plant, when the night is too short, flowering does not occur. Molecules called phytochromes play a major role in seed germination and plant response to seasonal changes. Phytochromes are leaf pigments that can be used to measure the length of night. In most phytochromes, the light-absorbing portion is photoreversible, meaning that it converts back and forth between two forms, depending on the color of light it's exposed to. In its PR form, a phytochrome absorbs red light and is converted to PFR form. In its PFR form, it absorbs far red light and is converted back to its PR form. PR is produced slowly in the dark, and PFR is produced quickly in the daylight. In daylight, there is a lot of red light from the sun. Some, but not much, far red light is also present. In darkness, there's neither red light nor far red light. PFR is slowly converted back to PR. It is the levels of PFR that are used in determining the length of night. Long day plants need high levels of PFR if they are to bloom. Short day plants need low levels of PFR to bloom. In long nights, lots of PFR is converted to PR. Long day plants flower when the day length reaches a critical period. This allows PFR to build up to a critical level, stimulating the release of flowering hormone. Short day plants require a long period of darkness, allowing PFR to fall below a critical level to flower. So how does that work again? Long day plants bloom when nights are short because less PFR is converted, resulting in high PFR levels. PFR then binds to a protein which acts as a transcription factor that switches on the gene for flowering. Short day plants bloom when nights are long because more PFR is converted, resulting in low PFR levels. PFR acts as an inhibitor of flowering in short day plants. To this point, we've only discussed plant responses to light. However, plants also respond to a number of other stimuli. But why do stems grow upwards and roots grow downward? You guessed it, gravity. Gravitotropism begins as soon as a seed begins to germinate. It's thought that a structure called a statolith helps detect gravity by settling to the lower side of the root. This, coupled with auxins inhibiting cell elongation on the bottom side of the root, causes the root to bend downward. Environmental stresses can also impact plant growth. If a plant is exposed to dry conditions, its stomata close, which slows transpiration. This stimulates the production of abscisic acid, which is also known as a stress hormone. Abscisic acid helps keep the stomata closed. Too much water can also be a stressor for plants. In this case, roots become waterlogged and face oxygen deprivation. This situation leads to the production of ethylene, effectively killing some of the cells in the root cortex. This creates air tubes which provide oxygen to the submerged roots. Temperature extremes can lead to the plant producing proteins that counter these effects. If too hot, heat shock proteins keep other proteins from denaturing, and if too cold, it leads to the production of antifreeze proteins. Plants can also respond to herbivory and pathogens. Defenses against herbivores include thorns, spines, trichomes, and toxic or distasteful compounds. Plants may also produce attractants that recruit animals that destroy herbivores. We will address these in more detail when we study community interactions. The plant's defense against pathogens begins with the intact epidermis and periderm. But open stomates and wounded surfaces from herbivores still leave the plant vulnerable to attack from bacteria, fungi, and viruses. If the pathogen invades the plant can mount a second chemical line of defense which can destroy the pathogen and prevent its spread to other areas of the plant. A virulent pathogen would kill the plant. An avirulent one mildly harms but does not kill it. These are much more common. Plants and most of their pathogens have co-evolved to where the pathogen gains enough access to its host, the plant, to enable it to perpetuate itself without killing or severely injuring the plant, much like a parasite-host interaction. Gene-for-gene recognition is a form of plant disease resistance where the pathogen is recognized by resistance proteins, coded for by DNA. If a plant does not have the appropriate R protein, the plant could become infected by the pathogen. Yet plants that do have the R protein can then trigger a specific defense mechanism like the hypersensitive response. This response causes cell and tissue death near the site of the infection, effectively restricting its further spread. Here's how this happens. The pathogen's effector proteins bind to a specific plant R protein. This triggers a signal transduction pathway. The plant cells release antimicrobial molecules and seal off the infected areas by modifying their cell walls. The cells then destroy themselves, producing lesions on the leaf. Before the cells die, they release a signaling molecule called methylsalicylic acid. This signaling molecule is distributed to the rest of the plant. Methylsalicylic acid is converted to salicylic acid, which initiates a second signal transduction pathway. The end result is systemic acquired resistance, where proteins are made that help protect the plant from further attack for a few days. Epidemics in the plant kingdom have had devastating effects on humans. Think of the Irish potato famine and the chestnut blight. Our current practice of monoculture leaves us vulnerable for another attack due to decreasing genetic variation in plant species. To help prepare for future epidemics, seed banks are being stockpiled that could hopefully contain genes that would curtail the next invasion. This concludes our study of plants. Can you see how life processes are similar across the kingdom? Botany is fascinating, and we've only scratched the surface. I do hope it's been helpful.